Hey guys, so for part two, we are still covering meiosis. We're going to cover metaphase one all the way through till the end of meiosis. We are still operating on the same standard and objective. You've already written those down, so don't worry about doing that today. Our essential questions are still the same. What's responsible for the diversity of life? And how is genetic information carried from one organism to its offspring? So we already covered prophase one, okay? Today we are going to start with metaphase one, okay? So during metaphase one, the paired homologous chromosomes, so instead of just one chromosome, we have a pair, okay? And they line up across the equator, aka the middle of the cell, okay? Just like we're the same thing is happening in metaphase one. It's just like as in normal metaphase, it's just pairs as opposed to just a chromosome. Okay. Remember, metaphase means middle. Okay. Anaphase one, during anaphase one, spindle fibers pull the homologous chromosomes pairs towards the opposite ends of the cell. Okay. Homologous pairs are moved away from one another. Okay. So, basically, we had two, okay, one and one, okay, each one's going to go in opposite directions and move away from each other, okay, so anaphase still means moving away. All right, telophase one, during telophase one, we still, telophase means two, so two nuclear membranes reform around the cluster of chromosomes, okay. All right, and then we have cytokinesis, just like at the end of mitosis, okay, where the cytokinesis splits the cytoplasm, okay, and then splits into two big cells, okay, and each new cell is called a daughter cell. That's what we call them, okay, they're like the baby cells, okay, two new daughter cells, and then the chromosomes unravel and decondense back into chromatin, okay. So the result of meiosis one, okay, this is what happens. We go from prophase one, they line up in the middle in metaphase, they're torn apart in anaphase. Telophase, we have two new nuclear membranes, and at the end we have two cells. Okay, we begin with one diploid parent cell, and we end with two haploid daughter cells. Okay, y'all have already covered diploid and haploid, so I'm just gonna be comfortable with those terms. Okay, so we go from one diploid to two haploid. Here's the start of prophase two. Okay, so as cells enter prophase two, their chromosomes consist of two chromatids. Okay, this is going to be just like mitosis. Okay, so chromosomes become visible. The chromatin winds up into our chromosomes, okay? They don't pair to become tetrads, okay? So they don't, they don't pair up into the pairs, okay? The nuclear membrane still breaks down just like in prophase and mitosis, but no crossing over takes place, okay? So we already had that crossing over. Okay, so the chromosomes have already swapped pieces, okay, to mix up the genes. All right, so that doesn't happen again. All right, in metaphase two, it's super simple to metaphase one. Chromosomes line up in the center. They're not in pairs anymore, so it's just a line of chromosomes. Okay, M for middle, same thing. Okay, they line up in the middle. Okay, except this is happening in two cells instead of one. Mona? Oh my God. All right. In anaphase two, it's really similar to anaphase one. The paired chromatids are separated by the spindle fibers. Chromatids move to opposite poles of the cell. Okay. So this is really similar to anaphase one, except for the fact that it's happening in the two daughter cells. Okay. A for away, same thing. All right. Okay. And telophase 2 in cytokinesis, the nuclear membrane still reforms in telophase 2, okay? And then cytokinesis occurs. The end result of mitosis 2 is 4 genetically different haploid cells called gametes, okay? We have, oh my God, we have 4 different cells, okay? They all have different genes. They're all mixed up because of crossing over, okay? And the, cells are haploid, but we call them gametes at the end of meiosis. God, I'm so sorry about my cat's butt. Good Lord. Gametes to zygotes, haploid cells produced by meiosis 2 are the gametes important to heredity. Okay, so after an egg is fertilized, the diploid zygote, okay, because it's now diploid because the egg cell, which was a gamete, and the sperm cell, which is a gamete, which each had, for us, 23, okay, will come together and make a zygote that has 46 chromosomes, okay? And they'll eventually, they'll undergo cell division by mitosis over and over and over again, and eventually form a new organism. So now we're on to our checkpoints. We have 4 or 5, I can't remember. So the first one, go ahead and answer the question, how does the process of crossing over lead to genetically different offspring? Go ahead and answer that question. Checkpoint 2, what are some of the differences between prophase 1 and prophase 2? So we're going to compare those. What's the difference between haploid and diploid cells? Checkpoint 4, I want you to list the phases of meiosis in order from start to finish. Okay, so including meiosis 1 and meiosis 2. List them all in order. And then lastly, you're going to compare and contrast meiosis 1 and meiosis 2 in a Venn diagram. Okay, so just like always, okay, we have meiosis 1 in the first bubble. Okay, then we've got meiosis 2 in the second bubble, and in the middle will be things that they have in common for both. Okay, so on your paper, go ahead and compare and contrast meiosis 1 and meiosis 2. And that is all I have for you today. Congratulations, we're done with meiosis. Good job.